Good morning everyone. Today we are going to start with amino acid metabolism. Here we have to study about transamination, deamination and decarboxylation. See, the general reactions of amino acids we are going to study. Transamination, deamination, decarboxylation of amino acids. Now, first we have to know about the metabolism of amino acids. Amino acids undergo transamination reaction and then it will undergo deamination and finally it will liberate the ammonia. The ammonia which is liberated, the ammonia group of the amino acid is utilized for the formation of urea, which is the excretory end product of protein metabolism. The carbon skeleton, so amino acid consists of an amino group and a keto group. So the amino group, it is the excretory end product, and the carbon skeleton of the amino acid get converted into a keto acid, which is utilized to generate energy, used for the synthesis of glucose, for the formation of fat or ketone bodies, for the production of non-essential amino acids. The dietary protein which you take or the body protein will get converted into amino acid. This amino acid is utilized for the protein synthesis or for the synthesis of the nitrogen containing compounds, and some part of this amino acid will undergo transamination and get converted into keto acid. This keto acid will give energy, will get converted into carbohydrate, get converted into fat, and can be utilized for the synthesis of non-essential amino acids. Transamination. So as the name tells, transamination. The transfer of an amino acid from amino group, from an amino acid to a keto acid is transamination. This process involves the interconversion of a pair of amino acid and a pair of keto acid catalyzed by a group of enzyme called a transaminase. So in general you can tell it is an interconversion step. The transfer of an amino group takes place and it is done by the help of transaminase enzyme. Now you can see here it is an interconversion. So we have an amino acid here, we have a keto acid here. The amino acid will give its amino group to keto acid. At the same time, the keto acid will get converted into an amino acid. You can see amino acid 2, and the amino acid without an amino group get converted to a keto acid. You can get alpha keto acid 2. By the help of enzyme transaminase, this is done by the help of PLP, pyridoxal phosphate. The same example here, it is an interconversion. Here you see the amino acid is there and a keto acid is there. The amino acid will give its amino group. So this amino acid will become a keto acid, and this amino acid will become a keto acid, and this amino group will be accepted by another keto acid and it becomes another amino acid by the help of transaminase. Okay, same example shown here. Here it is alanine. The amino group is given. It is accepted by pyridoxal phosphate. So pyridoxal phosphate will accept the amino group. It will become a keto acid pyruvate, and this pyruvate the pyridoxal which accepts the amino group become pyridoxal amine, and this pyridoxal amine will give its amino group to alpha ketoglutarate keto acid, and it becomes glutamate. Now, deamination. The removal of amino group from the amino acid as ammonia is called as deamination. It takes place by two steps: oxidative amination and non-oxidative amination. We will see one by one. Oxidative amination is the liberation of free ammonia from the amino group of the amino acid coupled with oxidation. So in simple you can tell liberation of ammonia, deamination, liberation of ammonia, oxidative with the help of oxygen. This takes place in liver and kidney. The purpose of oxidative deamination is to provide ammonia for urea synthesis. We know that urea is the end product of the protein metabolism. So to provide urea, this oxidative deamination takes place, and alpha keto acid for a variety of reactions including energy generation. You can see in the first step transamination is taking place, the transfer of amino group. In the second step, deamination is taking place, that glutamate. In the first step, transamination takes place where the amino group is transferred to a keto acid and it becomes a glutamate. In the second step, the same glutamate will undergo deamination and ammonia is liberated. Now, oxidative deamination. Yeah, oxidative deamination, you can see here what happens is that the first step, ammonia from the amino acids is transferred to alpha ketoglutarate to form glutamate. So we can tell glutamate is the collection center of free ammonia. This will undergo oxidative deamination to liberate ammonia, and by the help of enzyme glutamate dehydrogenase, which utilizes NADP or NADP+. Yeah, non-oxidative. So the liberation of ammonia without using oxygen. Some of the amino acids can be deaminated to liberate ammonia without undergoing oxidation. Non-oxidative deamination takes place by 3 steps, by 3 enzymes: amino dehydratase, amino desulfhydratase, deamination of histidine. These are the 3 non-oxidative reactions. One by one we can see. Amino acid dehydratase. So the common amino acid you can see down: serine, threonine, homoserine. These are the 3 amino acids which are having hydroxy group will undergo deamination reaction. The enzyme used is dehydratase, removal of ammonia, and they will get converted into respective alpha amino acids. Okay. Amino acid desulfhydratase. Example, the sulfur containing amino acid homocysteine and cysteine will get converted into will undergo deamination, desulfhydration, and get converted into corresponding amino acid. Cysteine, by the help of desulfhydratase, removes ammonia and sulfur, hydrogen sulfide. So you can call deamination, desulfhydration to produce pyruvate. Deamination of histidine. Histidine, by the help of enzyme histidase, removes ammonia and become urocanate. The metabolism of amino acids. Amino acid will undergo metabolism to liberate ammonia and other nitrogenous compounds. So the various steps involved in metabolism are formation of ammonia, transport and storage of ammonia, functions of ammonia, disposal of ammonia, and toxicity of ammonia. How the ammonia is formed? The amino acids, biogenic amines, and amino acids, biogenic amines, amino group of the purines and pyrimidine, and by the action of intestinal bacteria on urea will produce ammonia. So first step, formation of ammonia. Second step, the tissues produces ammonia, and even though tissue produces, in the circulation it is very low, because body has efficient mechanism for the transport of ammonia and immediate utilization for urea synthesis. Once the ammonia is produced in the body, it is toxic, so our body has a mechanism to remove the ammonia. It is removed in the form of urea. Yeah. Third step, transportation. The ammonia which is produced in our body is not transported as free ammonia. It is transported either as glutamine or it is transported as alanine. What are the functions of this ammonia? Ammonia is not just a waste product of the nitrogen metabolism. It is involved in the synthesis of many compounds in the body. They are involved in the synthesis of non-essential amino acids, synthesis of purine, synthesis of pyrimidine, amino sugar, and asparagine. The ammonium ions are important to maintain the acid-base balance of the body. The amino acids, the nitrogen is not only used for the synthesis of many compounds, the ammonium ions are also important for our body. They are required for the maintenance of acid-base balance of the body. Now, finally, so first it is in how it is produced, next it is transported, and then we have to study the disposal of ammonia, how it is disposed. There are different mechanisms of disposal from the body. There are three main it is there: aminotelic, uricotelic, ureotelic. What are these? Aminotelic. The aquatic animals dispose of ammonia into the surrounding by the process called aminotelic. Uricotelic. The ammonia is converted into uric acid and disposed. It is done usually in reptiles and birds. Ureotelic. For mammals, it is converted to urea. Urea is a non-toxic and soluble compound, and hence it is easily excreted. Now, what are the toxicities of the ammonia? So, if too much ammonia is there in our body, what can it lead to? It is harmful to the brain. Ammonia can accumulate in the body, results in slurring of speech, blurring of vision, tremors you can see, and can lead to coma and finally death. Okay. Now, if the ammonia level is elevated in our blood, what can you tell that condition? Hyperammonemia. It is a condition where elevated ammonia level in the blood. Impairment in the urea synthesis due to defect in any of these 5 enzymes in the urea synthesis can lead to hyperammonemia. Hyperammonemia can lead to hepatic coma, mental retardation, etc. Yeah. So, this is the metabolism of amino acids. Thank you.